Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year was 1983. The name, Barbara Mc-Clintock. The work, a landmark contribution to the understanding of genetics, the discovery of mobile genetic elements. To fully appreciate the brilliance of Mc-Clintock's insights, one must journey back through the intellectual pathways that paved her path. Her work drew upon a deep understanding of the intricate workings of the maize genome, a subject to which she had devoted decades of meticulous study. Prior to her groundbreaking research, the prevailing view of the genome was one of relative stability. Genes were thought to reside in fixed positions on chromosomes, inheriting a passive role throughout an organism's life. Mc-Clintock, a relentless observer and experimentalist, challenged this static view. The intellectual landscape of genetics in the 1940s and 50s, the period when Mc-Clintock laid the foundation for her later revelations, was in a state of thrilling flux. The discovery of the double helix structure of deoxyribonucleic acid, or DNA, by Watson and Crick, a monumental event, was still years away, but it provided an understanding of the physical basis of inheritance. Early work investigating chromosomal aberrations and the principles of linkage, although incomplete in its understanding of molecular mechanisms, provided the conceptual groundwork to understand the complexity of the maize genome. The concepts of mutation, recombination, and gene expression had all been established, but the true extent of genetic dynamism remained undiscovered. Mc-Clintock, by focusing her attention on the visible characteristics of maize kernels, such as their color and spotting patterns, developed an uncanny sensitivity to the minute changes reflecting underlying chromosomal rearrangements. Mc-Clintock's initial work, culminating in her seminal papers of the 1950s, focused on the observation of unusual chromosomal behavior in maize, particularly during meiosis. She observed that certain genes, responsible for kernel color and other traits, appeared capable of changing position on the chromosome. This transposition, she found, occurred at specific times and was often correlated with the appearance of visible characteristics on the kernels. This unexpected behavior indicated the presence of controlling elements, later named transposable elements, capable of both activating and inactivating genes. The key to Mc-Clintock's discovery lay in her meticulous and painstaking data collection. She crossbred maize plants for many generations, meticulously noting the color patterns of the kernels produced. The patterns were a direct reflection of the activity of the genes. The visual record of her work, the kernel patterns themselves, served as a primary source of information. She developed a deep familiarity with the genetic variations and chromosomal structures of her maize lines. Through this process, she was able to reconstruct the behavior of chromosomes to understand the underlying mechanisms. Her experiments involved the pairing of the maize chromosomes and mapping the expression of genes. The core tenets of her 1983 paper, though revisiting concepts developed earlier, provided a synthesis by describing and explaining the nature of mobile genetic elements. These elements, she demonstrated, could move from one location on the chromosome to another. She used the terms controlling elements to describe these mobile genetic entities. These elements were not simply inert passengers. They possessed the capacity to interact with other genes by jumping into or out of them. This resulted in a dynamic interaction between these elements and the surrounding genetic material, altering gene expression and resulting in visible phenotypes. The mechanisms underpinning this transposition were, at the time, only understood at a descriptive level. Mc-Clintock elucidated two key types of controlling elements: the dissociation DS element and the activator AC element. The AC element, she found, contained the genetic information necessary for transposition. It could move around in the genome on its own. The DS element, on the other hand, required the presence of AC to move. The relationship between the AC and DS elements helped explain the patterns of genetic change that she observed in the corn kernels. These visible patterns, meticulously documented, were key to understanding the dynamics of the maize genome. The specific techniques that allowed Mc-Clintock to make her extraordinary discoveries were a combination of classical genetics, painstaking observation, and insightful interpretation. Her work relied heavily on the analysis of the observable characteristics of maize kernels. This involved carefully noting the patterns of kernel color and spotting, and then correlating these patterns with specific chromosomal arrangements. She meticulously constructed genetic maps, indicating the location of genes and the positions of the controlling elements. Mc-Clintock also used cytological techniques to visualize the chromosomes. In this process, she observed the physical structures of the chromosomes at different stages of cell division. This was particularly critical for understanding the behavior of the controlling element during meiosis. The conceptual framework underpinning Mc-Clintock's work was revolutionary. It was a direct challenge to the prevailing dogma of the time, which held that genes were fixed and stable. Her work provided for a model of a dynamic genome. At the heart of her framework was the recognition that there were controlling elements that could move about the genome and modulate gene expression. This challenged established understanding of the roles of genes, chromosomes, and genomes. This model showed the genome as a tapestry continually being rewoven, not just as a rigid blueprint. The immediate impact of Mc-Clintock's work was significant, albeit not immediately recognized by all. The initial reception of her findings was mixed, with some scientists expressing skepticism. The idea of a dynamic genome that was capable of rearranging its own structure was a difficult concept for some to accept. Others, however, recognized the profound implications. Mc-Clintock's work provided for an alternative way of understanding genetic change and evolution. It opened up new avenues of research into the structure of genes. It stimulated researchers to look beyond the static view of the genome. The discovery also became a powerful demonstration of the importance of careful observation and a willingness to challenge existing paradigms. Subsequent studies refined and expanded upon Mc-Clintock's original work. Investigators learned the molecular mechanisms of transposition, detailing the steps by which transposable elements move from one place to another in a genome. The discovery of reverse transcriptase, an enzyme capable of making DNA from RNA, was instrumental in understanding the structure and function of one type of transposable element. It was found that retrotransposons, a type of element, use reverse transcriptase to copy their genetic material into the host's chromosome. Scientists understood that transposable elements are prevalent in all life forms. The discovery of transposition had widespread impacts. It was applied to diverse fields, including plant breeding and molecular biology. The study of mobile genetic elements provided scientists with new tools for manipulating genes in organisms. It has also provided insights into the causes of genetic disease. Transposable elements have been found to be a significant source of genetic variation. They contribute to the evolution of species. They can also disrupt genes and cause mutations. The discovery of transposable elements has expanded our understanding of the basic mechanisms of evolution. The reception of Mc-Clintock's work evolved over time. Initially met with some skepticism, her research ultimately gained widespread acceptance and appreciation. Her work was initially perceived as difficult to interpret, but later it became clear that she had unveiled a major principle of genetics. It took time for the scientific community to fully grasp the dynamics of the genome that Mc-Clintock illuminated. The award of the Nobel Prize in Physiology or Medicine in 1983, the same year as the publication of this critical paper, underscored the ultimate recognition of her brilliance. Numerous critiques and revisions of Mc-Clintock's work have emerged over the decades. Some criticisms focused on the complexities of maize genetics, others focused on the technical difficulties involved in studying transposable elements, especially before the advent of modern molecular biology techniques. However, the core tenets of her work, the existence of mobile genetic elements, and their role in genome dynamics, has remained robust. Subsequent research has built upon, refined, and expanded her initial findings, providing greater detail about transposition mechanisms. This work has highlighted the diversity of transposable elements. It has also helped us understand their role in disease and the work of Mc-Clintock was directly influenced by the earlier work of other geneticists. Our appreciation for the importance of visual observations and the meticulous analysis of genetic data was honed through her training at Cornell University, where she studied under the geneticist L.W. Sharp. She was also influenced by the work of Thomas Hunt Morgan and his students, who established the chromosomal theory of inheritance. Their research on fruit flies showed how genes are located on chromosomes. This provided essential background. Specifically, her research on the maize genome was also influenced by her colleague and friend, Marcus Rhodes, another prominent maize geneticist. Their interactions and collaborations provided the intellectual environment. Rhodes supported Mc-Clintock's pursuit of difficult problems. The collaborative atmosphere in the maize genetics community was a key factor in the discovery. The impact of Mc-Clintock's work on others was profound. Her work fundamentally changed the way scientists understood the genome. It impacted the work of several colleagues, including James Shapiro, who contributed to the theory of the dynamic genome. Her work also influenced Susan Wessler, who has made significant contributions in understanding the role of transposons in plant evolution. The concepts developed by Mc-Clintock have been applied in various fields, including crop technology. Her influence extends far beyond specific research fields. The story of Mc-Clintock's discovery provides a powerful example of the importance of intuition, persistence, and a willingness to challenge conventional wisdom. The very language that Mc-Clintock used to describe her discoveries, such as jumping genes and controlling elements, became iconic. They remain central to our understanding of genetics. Her legacy is a testament to the power of observation. She has profoundly influenced future generations of scientists. It continues to inspire a new era of geneticists dedicated to the study of the complex and dynamic world of the genome. In essence, Barbara Mc-Clintock's 1983 paper and its preceding works serves as a vibrant testament of the value of curiosity and the lasting nature of scientific innovation.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.